Hi, good afternoon.
Hey, Mani. Can you
You're mute, Dr. Armagam.
Yes. Hi. Can you hear me now?
Yeah.
Yeah.
Okay, great. Thanks for the invitation.
Oh, it's a pleasure. We've all read your work, and I'm sure the students were really excited to talk to you earlier today. And I think we'll have a lot of discussion as part of this colloquium as well. So
I hope
I
so.
think
I hope so.
Yeah. So I think we'll get started. Welcome everyone to the seventh IBSE Colloquium. So we've been doing this for a few years now, and where we have some really eminent speakers coming in and talking about some exciting work. And I think in the long list of those eminent speakers today, we have Dr. Mani Arumugam, who's a group leader and associate professor at the Novo Nordisk Foundation Center for Basic Metabolic Research and the University of Copenhagen. We are very delighted to have you. I'm sure many of you are already aware of his background and a lot of his research, but I will still take the privilege of introducing him. Dr. Arumugam leads the Microbiome Systems Biology Research Group. It investigates how health and diseases are influenced by our gut microbiome, and specifically how the gut microbiome interacts with host and environmental factors, as well as how we can therapeutically modulate the gut microbiome to improve health. I'm sure a lot of research at IBSE that's ongoing, which has overlap with this, and I think the students will be very excited to listen to you. And his group takes interdisciplinary approaches combining multi-omics microbiome data to study host microbial crosstalk. They also look at therapeutic approaches to modulate the microbiome. And he's got several years of experience, nearly 15 years of experience in this. And he's a principal investigator in several studies, lots of interesting grants. I'll not give put all the details in front of you, but you can look up his website. I think you all know that he's published some really excellent research. He's published about 53 research articles, with several of them in Nature, Science, and Cell, and an H-index of 32, with about 19,000 plus citations. So that's more testimony to how impactful his research has been, and how much it has pushed forward the science of the gut microbiome. He was, of course, listed as amongst the highly cited researchers in 2018, 19, and 20. So not surprising, but it's really good to see that. And I think you all probably got introduced to his work with the enterotypes of the human microbiome. And I think I'm sure the students always like to see, an author of a paper that they've read and cited multiple times in Flesh and Blood. So Dr. Mani, we look forward to hearing from you.
Thanks a lot for the kind words, Karthik, and I will start sharing my screen.
Yeah.
And I hope you see the right version of the screen.
Yeah.
Okay, good. It's a pleasure for me to talk to this audience. Brings back fond memories of my time in Chennai, just across the street. I was just telling the students I was interacting with an hour ago that my interaction with water, it moved over from fear to familiarity because of the IIT swimming pool, because that was the only swimming pool available at that time within a radius of I don't know how many kilometers from AC Tech. So thank you for offering your swimming pool to people like me to get over our fear. And also, I have fond memories of visiting your campus for your cultural festivals, etc. It would have been great if it was in person, so I would have had a trip down the memory lane. But we live in a strange world these days, so this will have to be. But hopefully, I will be able to visit and meet you all in person. Today, I want to talk about my favorite topic, which is the microbiome. I said analyzing the microbiome for trends, biomarkers, interventions, and integration. It's more a summary of the trajectory I've had in the past rather than what we are actively working only right now. So it will start from the very beginning of my career. And hopefully you will have questions that I can answer at the end of this. For those of you who may not know what the microbiome is, just to make sure that we are all on the same page, let me introduce this very briefly. And if you already know this, please bear with me. The human microbiome is just a collection of organisms that are not human, but microbes that are associated with our body. They live in, on, and around us. We are practically a walking zoo. There's trillions of microbes that we are host for. Only in the gut, there's easily 1 to 2 kilograms of microbes. And there's also speculations that this weight might be actually dry weight and not wet weight, which means a significant fraction of our weight is made of purely microbes. Especially one part of that, the gut microbiome, it constitutes the largest number of bacteria in our body. It's a major component. It's quite essential. We cannot really live without them. They help us in our daily life. They interact with the immune system and defend us against pathogens. The gut microbiome and the immune system, they constantly shape each other, and the gut microbiome is very crucial for early development. You might have read about what happens to C-section born kids because they are missing out on the early interactions with the gut microbiome, or the proper seeding of the gut microbiome. The gut microbiome also stimulates the immune system constantly. That keeps our immune system in check, more like training. Independently, they also act as a protective barrier against pathogenic organisms that might want to invade our body. That's about the immune system, and also on an energy balance perspective, they also help us metabolize a lot of different components of our food. They help convert dietary polysaccharides that we cannot digest ourselves. We don't have the enzymes for that. Especially the complex polysaccharides. They convert them into energy and convert them into short-chain fatty acids that, for example, the enterocytes can use directly as energy. There's also a role of them in synthesizing vitamins and then giving them to us. Today, you might consider this a moot point. You just have to buy vitamin supplements from a pharmacy and pop it in. But before that, if you think about a couple of 100 years earlier in human history, of course, that was not available, and there was only one source, which was diet, and the other source was our own microbes. The main reason we study these microbiomes these days is because they are strongly associated with several diseases. I am particularly focusing on the gut microbiome. So for the rest of the talk, that's what I'm going to talk about. Even if I just say microbiome, I mostly mean gut microbiome. Several diseases have been associated with the changes in the gut microbiome. I have listed some here. This is not an exhaustive list, and I've color-coded them based on broad categories. Some of them will make sense. Colorectal cancer at the bottom right, it's a cancer in the colon, and the gut microbiome lives in the colon. So obviously, it makes sense that they are linked. Obesity and diabetes on the top right. We just discussed that it could be the way they convert diet into energy. Maybe they give us more energy or less energy depending on who there is. Kind of makes sense. But look at the left side. There's associations between autism and gut microbiome, multiple sclerosis and gut microbiome, Parkinson's disease and gut microbiome. There's no reason, at least until now, we can think of very easily how microbiome might be affecting these diseases or might just be associated with these diseases. So a lot of work still remains to be done in understanding these associations, because so far all we have observed are associations. There is a change in the microbiome and there is disease, but we do not know what came first, what caused the change, or what caused the disease. And this directionality and causality still remains to be established because we do not know the molecular mechanisms behind these associations. So it's really difficult to say whether changes in the microbiome was caused by colorectal cancer, for example, or the changes in the microbiome were the reason for colorectal cancer. If we want to figure these out and understand the molecular mechanisms, we need to study the microbiota in great detail, and that needs a technique called metagenomics. That's because if we want to understand the role of microbiome in these diseases, for example, we need to understand the way the microbes and the host are interacting more at the molecular level. If you want to understand the microbes at the molecular level, of course, we need to understand their genomes. We need access to the genomes and the genes that they're encoding and the products out of these genes, both proteins and metabolites. But the bottom line is, for that, we need to understand to get access to the microbial genomes. The problem with gut microbiome is that most of the bacteria are difficult to cultivate. It's not impossible, but it's quite difficult. You have to spend a lot of time in optimizing culture media to culture many of these bacteria. And a lot of them are anaerobic, which means that you need special equipment to culture them in the absence of oxygen. This means that only a few genome sequences are available for the gut microbiome members. This number is growing these days, using a lot of different techniques. I'm not going to touch about that, but there's also a technique called metagenomically assembling genomes, which provides access to a lot more genomes than we had in the past. But from cultivating bacteria and getting a clean genome from a single organism, we still have only a few. This leads us to a requirement that we need to characterize the genomes of the gut microbiome, not by taking one by one and isolating them and culturing them and then sequencing their genome, but more as a whole community of organisms. Can we characterize the entire gut microbiome without the need for culturing them, because it's really difficult to culture them? And a lot of them have interdependencies between each other, which means that it will be difficult to culture one of them alone. So the best bet will be to look at them all together, and that's what metagenomics as a technique offers. It provides us a culture-free genomic snapshot of a given environment, let's say the gut microbiome. With that quick and short introduction about microbiome and metagenomics, which is the tool that we traditionally use to study the microbiome, I want to walk you through some of the discoveries we've had in the past. To start with some trends in the human gut microbiome we have observed using metagenomics. To understand what's going on in the microbiome, the first question we usually ask is who is there in our microbiomes? And that question can be answered at different levels. We might mean which species are present in our microbiomes. We might mean which different genera are present in our microbiome, or which phyla are present in the microbiome. These are taxonomic ranks at a different level. You can compare that to, for example, origins of, not origins, but affiliations of humans. We can say which country are you from, and then which state are you from, and which city are you from, which street are you from, and every question has a different level of resolution. And you can think about kingdom, phylum, family, genus, and species as these different affiliations that are increasing taxonomically in resolution. You will have to probably tilt your head a little bit to read what is written. This is a single sample from a Danish individual that was sampled more than 10 years ago, where we identified which different bacteria are present in them and in what quantity. You can see, for example, that in this individual, I don't know if you can see my mouse, there's a bacterium called Prevotella copri. It was very high in this individual. probably about 20% was of this individual's gut microbiome was just that species. And there's a lot of other species, and that's just in one individual. So this is kind of how we answer this question: who is there in our microbiome? We have a list of bacteria we know, or an archaea that we know of, and we map against these bacteria and find out which of them are present. We can scale that up to multiple individuals. If you ask the same question, who is there in our microbiomes for a lot of different people, then you will have an answer like this. Now it's rotated, so you can read the names here more easily. But you also have much more entries here because across the entire cohort, there's much more species that are present. And you have names of these individuals and that are listed here. And the names are also telling you something about these individuals. There are these 4 individuals who are infants from Japan. These are adults from Japan, and these are old people from Italy, adults from France, Denmark, and America. Already by looking at something like this, where this is a phylogenetic display of how these different bacteria are related to each other. Each color represents a different phylum, and species that are close to each other are closer in proximity here in this tree. So it's a branch length free phylogenetic tree, if you will. There are some trends already that you can see here. I'll start with one. These 2 here at the top, Methanobrevibacter and Methanosphaera, they are archaea. They're not bacteria, but they are archaea. You can see that in Americans, there's a little bit more of these archaea compared to Europeans and Japanese. Of course, with only 2 samples, you cannot make strong conclusions, but at least you can see that it's highly represented. You can again check something else, which is that this region of Bacteroidetes, which has a lot of different species from the genus Bacteroides, you see many of these different species present in all the adults and old people, but there's a conspicuous absence of these bacteria in infants, and that is normal because infants have a different microbiome composition, and they don't get Bacteroides early in their life, but instead they have bifidobacteria. If you see here, all the infants have at least some amount of bifidobacteria, except perhaps this one individual. And these are some of the trends that you can get just by looking at what kind of bacteria are present in different individuals. Using the exact same data that I showed you, we identified that gut microbiomes have preferred compositions, and they're not completely random. It's not that you can come up with a random number generator that tells you X% of Bacteroides, Y% of E. coli, and you put these numbers together, that will be a microbiome. Well, that will be a virtual microbiome, but that may or may not exist at all in reality because they have preferred compositions based on the interactions between them and the balance between them. Moving from species, we went on to genes and started counting how many different genes that our microbiome carries. And there, in our first study, we cataloged 124 individuals from Europe, from Spain and Denmark, using more than 0.5 terabytes of metagenomic sequences. We identified 3.3 million genes in these individuals. This was aptly called our other genome when this was published in 2010. Later, we expanded that with more than 1,000 individuals, now also including subjects from America and China, where we now, this is the latest catalog that we traditionally work with 10 million genes that are coming from the human gut microbiome. If you compare that with probably about 100,000 genomes that the host carries, that is our genome carries, this is a huge number that is also very dynamic and also has a lot of scope for modulation. So that tells us something about why this is important. So we went from species to genes, but we can also go into another level of resolution, which is strain level variations at the within these species, which are driven by single nucleotide polymorphism, SNPs. What we did was to look for SNPs inside specific bacteria, again from a group of individuals from America and Europe. What we found was that if you compare, these were followed over a year. If you compared how SNPs were maintained in the microbiomes in the same individual over time, compared to how bacterial species were maintained in the same individual over time. These are the two comparisons you're making. Within the same individual, let's start with the bottom part here. Within the same individual, in red, you see how much similarity there is between two samples of the same individual and over time. And in red, you see that within the same individual, well, they're similar. The similarity is on an average about a little over 0.8. But if you look at how similar different samples from different individuals are, that number is even slightly higher. It doesn't mean that other people are more similar to us. It just means that there is no real signature. In terms of what species are in you over time, you can really differentiate you from others because the microbiome is very dynamic. But instead, if you're asking the question, how are the SNPs maintained in a given species that I carry over time? There, the role is reversed. There's a very clear signature that within the individual in red, SNPs are maintained pretty close to 1, which is the maximum. But between individuals, it is lower than what it is when you compare within the individual. Again, this number is the number in blue is also high. That's because we are mapping everything within one species, so you automatically expect the number to be high. But within the same individual, you actually carry your same strains over time, and that's the conclusion of this. Individual specific strains, they are stable up to a year because that's the maximum time limit we had in our sampling. Which means that my E. coli strain, if all goes well and if I'm healthy, because these are all healthy individuals we sampled, while I'm staying healthy, my E. coli strain will remain with me for a long time. You can actually get the signatures by its SNPs, and your E. coli strain, even though it is still E. coli, will be slightly different, carrying different mutations. So we went from species to genes and to SNPs to identify different trends in the human gut microbiome. I'll now walk you through maybe a story of identifying biomarkers in human gut microbiome. This is going to be just one story, and that was a story of biomarkers in colorectal cancer. We had performed a metagenome-wide association study for colorectal cancer diagnosis. This was in collaboration with BGI, Beijing Genome Institute, and also the Chinese University of Hong Kong. The hospital at the Chinese University of Hong Kong recruited 128 individuals, all from Hong Kong. And we performed shotgun fecal metagenomics on these individuals, 74 cancer patients and 54 controls. We performed an association study and looked for differences between these groups. And a lot of times, quantifying what bacteria are present in a sample, it's very difficult to agree on what's present, what's not, and what's the abundance level, etc. So what we did in this case, was to perform it using 3 different methods and only look for signatures that were preserved across these methods. And what we see here is that there are 3 different bacteria that were clearly associated with cancer status, no matter which method we use. These are the 3 methods listed on top. 2 of them were enriched in cancer patients. One of them was Parvimonas micra, and the other was Fusobacterium nucleatum. And one of them was actually depleted in cancer patients, so it could be considered a marker of health. Fusobacterium nucleatum already at that time was a well-known bacterium associated with the colorectal cancer, but Parvimonas micra was relatively new. We were the first one to highlight it, and in our hands, it was even a stronger candidate than Fusobacterium nucleatum for diagnosis. We also looked how they were interacting with each other. Of course, statistically, we never observed physical interaction, but statistically, how well are they correlated using a correlation network? Most of the members of microbiome that were associated positively with colorectal cancer, they were all tightly connected. They were all correlating with each other, which means that they all went up together in during cancer. Whether this was before cancer or whether this was after cancer, like I said, causality is still not clear when it comes to colorectal cancer. But our focus was, as I said, towards cancer diagnosis using microbiome. So we don't go beyond that. We don't try to figure out who's causing cancer, but we are focusing more on can we detect cancer using these microbes. What we wanted to do then was, if we want a real diagnostic kit that can detect cancer, we need something that can be used in a clinical lab very easily. And usually that's a gene sequence that you can do, you can measure by PCR, not the entire microbiome for which you need either 6-ns sequencing or short-term metagenomics. That will be too expensive and too complicated for a clinical diagnostics lab to perform. So we were thinking whether we can identify biomarkers that we can just qPCR up and measure. For that, we started with a large gene catalog that was available at that time, which with 4.2 million genes from the gut microbiome. From that, only half of them were relevant for our samples, which means from the 128 individuals that we had in our cohort, half of them were not even present. So we restricted ourselves to these genes. And from this, we identified about 25,000 genes that were associated with colorectal cancer. Then we used a machine learning method called MRMR, minimum redundance, maximum relevance. And using that, we identified 20 non-redundant biomarkers. The way it works is that it looks for one strong biomarker first, and then look for a second one, which is orthogonal, which uses complementary information. And a third one will be orthogonal to both the first two, and the fourth one will be orthogonal to all three. So each of them is iteratively a non-redundant biomarker. With 20 of these biomarkers, we managed to separate the cancer patients from the healthy controls very easily in our study. To also show that it's a cancer-specific signature, here you see all the cancer patients are on the higher end of this of the abundance of these genes. So on the x-axis, you have just the log abundance of these 20 genes summed up. If you sum them up, they are higher in cancer patients compared to healthy controls. When you do that for type 2 diabetes patients and IBD patients, the separation was not that clear, which means that this is not a general gut microbiome imbalance signature, but it's a colorectal cancer specific signature. Then we thought, well, we have a very nice set of 20 genes. Let's diagnose cancer. Let's just qPCR some of these genes and see if that is enough to diagnose cancer. For that, we had access to a new cohort of 47 patients and 109 controls. Of course, this was an independent validation cohort, but from the same hospital. We picked 4 genes out of 20. 2 of them were enriched in cancer patients, and 2 of them were enriched in healthy individuals. And we measured whether the index, which is the sum of the log abundance of these 4 genes, was separating patients from controls. And unfortunately, it was not. On the right, if you can see here, this is a log curve that shows how well the same 4 genes can classify somebody as having colorectal cancer or not. The area under the curve is 0.73. It translates roughly to 73% accuracy. It's not exactly accuracy, but it's roughly 73% accuracy. But the true positive ratio was quite low, only 50%. That didn't really work well, especially considering how strong they were in the original cohort. But that tells us something about independent validations. Normally, when we train our machine learning methods on a given cohort, it will nicely work, but then when you translate it to an independent cohort, it may or may not work. And this is what we were hit from. So that definitely did not work well, and we went back to the drawing board and start thinking, what can we do to improve the applicability? At that time, we also had access to 3 other studies that were published. So this is the discovery that we had. We had 20 genes from our Chinese cohort. They're all shown here in color. Those that are green are control enriched in healthy individuals, and those that are red are enriched in cancer patients. We then measured the exact same 20 genes in validation cohorts that were just published around that time. The Danish one was internal. We had access to a Danish cohort. It was a very small cohort with only 40 individuals, but for validation, it is not bad. 4 out of our 20 genes was validated in the small Danish cohort. Then we had access to an Austrian cohort that was published around that time. And 5 out of our 20 were validated there. And we had another cohort that was from French individuals. Again, 5 different genes were validated. So you can probably see a consensus emerging that across all the validation cohorts, there were 4 that were strongly validated. That tells us that cross-cohort validations are also important, because if you remember, we went from 20 and randomly picked 4 genes. But instead of picking those randomly, if we had done cross-cohort validations and then selected them, maybe we would have had a higher chance of success. We wanted to evaluate if that is true. So what we did was to pick 2 out of those 4 genes. And these 2 performed wonderfully. When now we went back to our Chinese validation cohort, where the 2 genes, if you qPCR them up, they clearly separated the controls from the cancer patients. You might think that, oh, well, there's a lot of them here that are not 0. So what, maybe I should explain the y-axis. At the bottom here, this is all practically absence of both the genes. That is this group. And here, at least one of them is present, maybe both. And if you look at the cancer patients, there's 47 cancer patients. There's easily about 30 where at least one of them was present, or in some, both were present, and a few where these were absent. So of course, that is likely that some of the cancer patients didn't carry these genes. But if you look here, around 109 controls were tested. Only a handful, less than 9, were positive for these genes. More than 100 controls did not have this. So it was a very clear signature that these 2 genes are present only in cancer patients. And the area under the ROC curve was very strong, 0.84. And this told us that this was a very good way of potentially diagnosing colorectal cancer. But of course, this was done in one cohort. So we then started thinking about it in a meta-analysis point of view. We collaborated with the group that published the French study, and also they published an American study from the same lab. And it was the lab where I did my PhD. So we went back and collaborated with them with access to 4 cohorts that were published and a new German cohort that were not published. I will go through this very quickly. Of course, when you look at 5 different cohorts where different hospitals sample different people using different procedures, there will be clearly batch effect, and there were batch effect. But even beyond the batch effect, there were some really true biomarkers. On the x-axis here, this is variance explained by study. Each of these circles is one bacterial species. And when the bacterial species is placed higher on this axis, it means that that bacterium changes purely because of which study it is from, which batch it is from. But on the y-axis, that's variance explained by cancer. And any bacterium that is higher in this axis, higher meaning going down because the axis is this way, those are biomarkers whose variants are primarily explained by cancer, but not necessarily by batch. So you want them to be around here, very low on batch effect, very high on cancer effect. And that's where many of the known bacteria that were from our original study, if you remember Parvovirus mica and Fusobacterium nucleatum, they were all here. there are some other species that were also part of the correlation network I showed you before. So this told us that there is true potential biomarkers, and we investigated them further. What we did was to train a lasso model for classifying cancer in one cohort and evaluated how well it will perform in a completely other different cohort independently. So training in France and testing in Austria, our area under the curve was 77%, 0.77. Training in France and testing in China was 0.84. So on an average, we had a reasonable accuracy of prediction. And this was all based on microbiome species. For testing whether functions inside the microbiomes are useful, we repeated it on functions, and we got similar results. So both the species and these functions they carry were predictive enough of colorectal cancer. Again, we cannot say anything about whether these caused cancer or whether they were a result of cancer, because this is purely an association study. What we also found out was that instead of transferring training from one cohort to another, what if we train on multiple cohorts and then hold one out and validate on that? We got an improvement of easily 5 to 10%. When you hold out France, train on all the others, and then test on France, compared to the one-to-one training before, the leave one study out and prediction was much stronger. So that was a little tour on biomarkers. So maybe I will now switch on to some studies we did with interventions on the human gut microbiome. Actually, it's only one study, I believe. Let's see. We performed an intervention on healthy individuals. What we wanted to do was how to investigate how do microbiota recover from an intervention. And in this case, we practically dropped the bomb. We recruited 12 healthy male medical students in our medical school, and we gave them all 500 milligram of meropenem, 500 milligram of vancomycin, 500 milligram of gentamicin. We mixed them all in orange juice, and we gave them this juice every 24 hours for 3 days. So it was oral gavage. And we collected fecal samples before this started at baseline. And after 3 days of treatment, we took a fecal sample on day 4, and then again day 8, day 42, and day 180, just to see how the microbiota are recovering. We started looking at how the microbiome composition was changing over time. And what we found was that, especially in these healthy young men, the microbiome was quite resilient. I will walk you through this. The red ellipse explains the 95% confidence interval of day 0, the baseline. So all the samples are here. And from here, when we put them through the intervention, which is 3 days of heavy antibiotics, and mind you, this concentration of antibiotics we gave them is much higher than regular antibiotic doses, because this was an attempt to completely kill the bacteria in their gut. But even though that's what we wanted to completely eradicate, they were not eradicated. They just moved to a different place in the microbiome composition space. The blue ellipse is the 4th day microbiome composition. So it was a huge jump. But then slowly over time, by day 8, they had moved in their composition. By day 42, they are moved again in this purple composition space. And day 180, they are in green. The green ellipse is a bit wider, but the green ellipse and the red ellipse are quite overlapping. It practically means that they got back to almost where they started. So these young healthy men, their microbiomes were quite resilient. Some microbes did disappear. If you look at the richness of these microbiomes, after 4 days, on an average, they had 150 species before, and after the 3 days of intervention, they lost half of their microbes. They stayed low, but then over time, they came back to a reasonable number. And you can see that there is still a decrease, and that's why we are saying maybe some microbes disappeared, but not completely. Somehow they bounce back. What was very interesting was that we also measured through the shotgun metagenomic sequencing data, we also measured how antibiotic resistance or carrying antibiotic resistance changed the odds of survival for individual bacterial species. For some, it was quite interesting. If you had resistance to glycopeptides, then the odds of survival for that bacterium was actually lower. We still don't fully understand why. But colonizing after the event, if you are a new bacterium coming in to colonize the gut of this individual, and these are bacteria that were not present in the baseline, if you had resistant to aminoglycoside, which was one of the antibiotics that we used, then your chances were very high. This part is kind of explainable, well, not explainable, but it can be said it's expected. Well, there's still aminoglycosides resting left over in your gut, so having resistance increases your chances of colonizing. But for example, here, survival in the gut when there is glycopeptides given, but survival was lower when you had resistance. We still didn't understand exactly why that was the case. Well, that's how science goes. Sometimes you don't understand everything. Why do they recover nicely? Maybe 3 days was not a long time. Normal antibiotic courses are a week long, sometimes a couple of weeks long. Retrospectively, it was a very highly controlled human experiment. We didn't just go to a random population. We recruited healthy Danish male medical students, probably of similar age because they're all in medical school, probably similar lifestyles, probably eating from the same cafeteria at the university. All this probably links this to a very controlled experiment. Potentially also their healthy dietary habits were helping. But that's at least how far we have gone in interpreting them. But the signatures of how interventions change the microbiome, in this case which was antibiotics, were quite interesting for us because the healthy microbiome did bounce back to where it was before. So the last part, which is some unpublished work and ongoing work that we are doing right now, integrating multi-omics readouts from the human gut microbiome. We use a in vitro fermenter system to study the human gut microbiome in a very controlled manner. We have a three-stage system where food and pancreatic juice and acids go into a compartment that represents stomach and small intestine. And from here, the culture is transferred to a proximal colon, where the pH is maintained similar to the proximal colon. So low pH here, medium pH here, and then the distal colon, it's neutral pH, and then the culture goes out as effluent. And we feed the system every 8 hours, so we get 3 meals a day. And it's a very nice way of studying the microbial ecology because we can get the samples anytime we want at very regular intervals, and this is not something you can get from human subjects. With this system, we can run 4 experiments in parallel. So what we do is when we start the experiment, we seed the culture with human gut microbiome with fresh fecal samples, introduce both here and here. And then the experiment starts. The food is processed here and transferred here, and the microbiota process this food and then transfer here, and then the microbiota again process this and kicks them out as effluent. But we can provide different food in the 4 different conditions, which is what we did. We had a controlled diet, which is a balanced diet, or a high fiber diet, or high protein diet, or high fat diet. And we followed this experiment for over a month. Well, actually, exactly a month, 31 days. The fecal inoculum, in terms of microbiome species composition, was here. On day 1, all the 4 different compartments are here. And then they started moving slowly. Day 3, day 5, and then you can see that there is a divergence starting. High fiber and control are here. High fat makes a huge jump and goes over this space, and high protein stays separate. So diet, in this case, just define nutrients that we fed to the system, they clearly shape the microbiome composition. With that, I will introduce you to my team. Of course, all the work that I showed here, except the early work done when I was a student, all the recent work was done by the talented members in my group. This is just the sample of who we are. And also thank you for listening to me. And I also want to acknowledge a lot of people who were involved in this work, past members of my team who are now moved on, and collaborators from University of Copenhagen, from Beijing Genome Institute, from Embu, where I got my PhD, and from the Chinese University of Hong Kong, where the colorectal cancer study was done. And with that, I will stop, and I will be happy to take questions.
Hey, thanks a lot for a wonderful talk. So I request all the attendees to post their questions in the Q&A box. I'm sure you have a lot of questions, and I can even maybe bring some of them as panelists, and they can voice their questions. But yeah. So very interesting. So obviously, lots of exciting work that you showed us. I was, of course, quite intrigued by your very last piece of work where you talked about, the metabolic pathways involved and so on, and how the the interplay between the diet and the microbiome. So have you also looked at this in the context of, say, cancer? Where are there like metabolic signatures of the microbiome and things like that? I'm not sure. And consequently, how again diet plays a role in cancer and things like a longer story of that sort essentially.
It's an excellent question. We didn't analyze it part of our study because we did not have access to metabolomes. But there was a study published in 2018 or 19, I believe. It was a Japanese study led by a former colleague of mine. So we studied in Embu together in Per Borg's lab. He went back to Japan, and I moved to Copenhagen, and he published on a Japanese cohort where they had both metagenomes and metabolomes. And they did find some signatures on both metagenomes and metabolomes. And they even found very specific signatures of metabolomes. In our study, by looking only at the functional capacity of the microbiomes using metagenomes, we had a link to bile acid metabolism. And bile acid metabolism usually goes back to meat and high protein diet, and they had some signatures on functional microbiome function and also metabolomes as well. Ultimately, a lot of them go back to diets, and this we already know. High fiber is probably going to keep us safe from cancer when everything else is balanced. And a lot of meat is probably not a good idea. A lot of processed food is probably not a good idea. So some of these were coming up, but of course it's not crystal clear. Again, all these studies were association studies. So what we can derive from this, we don't know yet. But on the other hand, what I can say is we have some plans to look more mechanistically. And that's also why we started these in vitro studies. So now we have products of micro.
Right. Really exciting. And what is your? Have you looked at any Indian data so far? There seems to be very little of it out there. So, what are your thoughts?
Okay, well, this is unpublished work. It's just been accepted in genome medicine. Well, officially not even accepted. It's in principle accepted. So I cannot say much. But it's a diabetes study comparing Danish and Indian individuals, where we do find some signatures of type 2 diabetes, but it's really difficult to find a global signature that is valid both in India and Denmark. For example, in colorectal cancer study, it was very clear. It was the exact same species that were showing up in cancer, both in India, both in the European populations and the Asian population, well, also in the American population. But that was not the case in type 2 diabetes because it's much more dynamic. But what we saw was that there was a huge difference between the Indian and Danish population in terms of the microbiome composition. And that's not new. Dr. Yogesh Chauche, he's already published on the composition of the Indian microbiome and how it is different. And some of the signatures we could recapitulate as well. But it's still only 16S. We haven't had access to shotgun metagenomes from India. But there is a new initiative, the Indian Microbiome Project that Dr. Yogesh Chauche from Pune is leading. They are going to generate a lot of shotgun metagenomics data from Indian microbiomes. So something to look forward to. I don't know if I will have access to that data directly, but I know him, so I might hear about what is going on with that.
Okay.
But we are very well. It's very different. Indian microbiome looks very different, at least in our cohort. Even though we had sequenced in the exact same facility, we had synchronized our sample collection and DNA extraction protocols. We tried our best, but they were very different.
Okay. So I think there are several more questions. So I think I'll quickly switch over to that. Maybe Manik, do you want to ask your question? You can just unmute yourself and ask. Because you're a panelist. So.
Okay. Hi, this is Manikandan. I'm a faculty at IBSE as well, and so a very interesting talk. So I think the main questions I had was in your multi-omics study. So which is if you only have resources to generate one of the types, right? Have you understood the data like that? We need reference studies such as yours to generate multiple types. But in the end, if you want to only generate one type that will have information about other types, which one is there such a thing, and which one would you generate?
It's an excellent question. We don't have an answer yet. What we have is in vitro data. We will try our best to figure things out with that. But I also have access to human data where metagenomics, metatranscriptomics, and metabolomics, and even metaproteomics are generated from fecal samples. This is part of a European consortium where I lead the multi-omics integration work. And that's also work in progress. The project started only last year. Currently, the most information that we have, that lies in metagenomic data. We understand the genomes better, we understand the genes better, we can annotate the genomes and genes and potentially predict what functions they might be doing. So it's known territory. Metatranstartomics is similar, only the expressed part of it, but it's a bit difficult to interpret it if all you have are fecal samples. The metatranscriptome will be telling you about what is happening in the feces, but that's probably 12 to 15 hours away from what is happening in the gut. And 15 hours is a long time for bacteria to entirely change their metabolic program depending on what nutrients are available. So fecal metatranscriptomics may not reflect the actual metatranscriptomes of the gut microbiome. So for now, we are focusing a lot on the fecal metagenomes because at least the presence of bacteria will tell you something. And using that as the core and everything else is surrounding that, mapping it back. What the metatranscriptome, these genes, which species they belong to, these metabolomes, these metabolites, who could be producing it, and the metaproteomes, which bacteria is producing these proteins and secreting them. So we are using metagenomics as a center. And that's not because that's the most important. So it's not a direct answer to your question. It's not because it's the most important, but it's the one where we have the most information. It's the most usable as of today. But the more data we generate, the better we will understand. And obviously, if you ask me today, what's more important? Of course, the orchestrators of the interaction between the host and the microbe, which is metabolites that are produced and secreted by them, and the peptides and proteins that are produced and secreted by them, those are the ones that are interacting with the host. So of course we need them. So metabolomes and metaproteomes, we definitely need them.
Okay,
but we are not there yet to understand them properly. If you find a metabolite, was it really produced by the microbe or the host, or is it coming from the diet? How would we even know, right? So
Yes.
it's very complicated these days, which is also why we kind of like our in vitro setup. Well, the host is out. I mean, we are studying the human microbiome. It might be considered idiosyncratic. Why do you take the human away from human microbiome and you're saying you're studying the human microbiome? You know what, as idiosyncratic it might sound, sometimes taking the host out is very important to understand what is going on. So in vitro studies might actually tell us something about how the microbes are transforming diet into different compounds, and that could be and how that could be interacting with the host. So again, not a direct answer to your question. Right now, practically the most useful is metagenomic data. Ask me maybe in 3-4 years, I might give you a different and updated answer. But thank you for your question. It's a very
So
interesting.
it's an informative answer. So I guess a longer answer I appreciate more than a direct one-worder. So the technical, I had another quick technical question is, there may be some abundant species in your sample. So how do you do any enrichment step to exclude certain species or focus on certain species to make sure that your metagenome or metatranscriptome data is not dominated by that one species and is representative of the concentrations of all the species?
There's not much you can do with, for example, studying a fecal sample. You get what you get, and you will get the abundances of the fecal sample or the fecal microbiome. And we have an internal project where we are trying to normalize the abundances at the DNA level. It's a protocol that one of my PhD students is working on. Can we enrich for rarer microbes and kind of partially deplete the most abundant microbes so that you see more? Because if your sample had 90% E. coli, you sequence a million reads, and 900,000 is E. coli, you sequence the same thing again and again. I think that's your question, right?
Yes,
Um,
yes.
but we are working on a protocol. It's not fully optimized yet. It seems to work. We had access to we bought a commercial synthetic microbiome community where the abundance is log scale distributed, 10 to 1 to 0.1 to 0.01. It works in a way to normalize it a little bit, but it's not a flat line yet. Maybe there is scope to optimize it, but that's all at the DNA level. If you take the microbiome in a community, and if you want to normalize it, you will have to manipulate it. There are some insights that we have right now. It's unpublished, and I'm not sure I want to go into the details of this, but if you want to write to me personally, I'm happy to give you some hints on it, because we're trying to hopefully get that published in a couple of months. So the short answer is you can enrich for rare species. We have recovered species that are not even detectable in the fecal sample, but we have cultured and managed to get the full genomes of them. I won't go beyond that because it's still unpublished and I don't want to reveal too much detail.
Sure. Yeah. Okay. Thank you very much.
So I think there are some questions in the Q&A box. So maybe I'll read out the first of them. So there's a question by Revati Ravishankar which talks about which is the most predominant microorganism in the gut, and now how does it differ from other microbes, and what is its percentage composition?
I will take you back to the enterotypes. So it depends on who you are. When we published the enterotypes first, we said there are 3 in our cohort, but of course it's not the final set. That's at least how we presented it, and it is true that it is not the final set. Now we have 4 enterotypes in the human gut that has been recently clarified. And that was my co-first author from that study, who is running his own lab in Belgium now. Depending on who you are, which enterotype you belong to, if you're a Prevotella type, you will have a very high abundance of Prevotella, and that could even be 40-50% of your microbiome. And among the Prevotella species, Prevotella copri could be a dominant one in many individuals, but it doesn't have to be Prevotella copri. There are other Prevotellus as well, non-copri. which haven't been named clearly. But if you're a bacteroides enterotype person, there are two different bacteroides enterotypes. One where it's purely bacteroides that is very high, and the firmicutes, especially the Lactobacillus, are low. And then there is another group which is bacteroides is high, but there's also Lactobacillus members present. Lactobacillus is a family consisting a lot of short chain fatty acid producers, all most considered good for you. So when you have both balanced, then you have a little less bacteroides because some of the space is taken by the Lactobacillus. And that's reasonably abundant one. In my experience, looking at the one Indian cohort we had access to, the abundance of Prevotella was higher in India than in Denmark. Again, this is unpublished, so you shouldn't go around telling everyone I said that. At least please wait for a month because I think it will be out by then. For example, Indians have more Prevotella. So the Prevotella enterotype in India will have higher amount of Prevotella than a Prevotella enterotype person in Denmark. The average person in India might actually have the same amount of Prevotella like the Prevotella enterotype person in Denmark. That's because there's more fiber in our diet in India compared to the diet in Denmark potentially. So a lot of it is diet driven. So again, a circular answer. You're all asking great questions for which there's no single one word answer. Depending on who you are, you might have 40% Prevotella copri or 30% Bacteroides vulgaris or uniformis, or if you are a Luminococcaceae enterotype, the most abundant will probably be 10 to 20% only because the Luminococcaceae enterotype, it's much more flat. There's a lot of different members. They're not overtaking, not a single one is overtaking, but a wide variety of firmicutes are taking over your cup. I hope that answers your question.
Yep. And there's one more question. So what strategies should be used to integrate metagenomics and metabolomics data?
I don't know yet. If I knew that, we would have published it already. All I can say is, well, people have already tried to analyze it. In America, the Human Microbiome Project Part 2, they have generated matching metagenomics and metabolomics data. And others have done that as well. The go-to guy for most cases is, well, take all your species, take all your metabolites, correlate them with each other, and whatever is correlated, then that is tightly linked, and let's see if the correlation networks tell us something. That's a good starting point. And when I showed our model where we integrate things, maybe I will just go back a couple of slides here. We are not very different. So our original model for now, it starts correlating each of these connections. The edges are drawn based on correlation. So we are also correlating metabolites with metatranscriptomes. So we are not also immune from making that mistake. But my hope is that we can go beyond correlations and start linking things more mechanistically. Don't ask me how yet. We have some projects which use deep learning to connect different features rather than just correlations. And this again is ongoing work. We haven't finished that yet. Today, if you ask me, I don't know. The only thing we know that can give us something like what you see on the screen is correlations. But I think that's only the beginning. That shouldn't be the end. I hope that others and us will hopefully come up with better ways of integrating metabolomics data with metagenomics. And we have a PhD student who is working on monocultures and predicting how we can link metabolites to bacteria using monocultures, and then see if this can be applied to microbiome setup. And this is again ongoing work, and hopefully this will be maturing in a year or so.
Okay. So there's a question by Dr. Komalapriya. Komalapriya, you can just unmute yourself and ask your question.
Thank you, Karthik. It's a very interesting piece of work, Dr. Mani. So I just had a couple of quick questions. So the first one is, it's very nice to, it's always an exciting topic when you talk about microbiome. And I understand that you don't know what causes what, and this is always an issue. But do you see any time in future you can use this data for predicting diseases? It I don't know. What are your comments on this? Do you think you can do it?
It's a good question. I do believe so. It doesn't matter whether microbiome causes the disease, but if you can see a change in microbiome before the traditional diagnostic tools, then we have an advantage. So we can use them to diagnose, even if they are not causing the disease. And I can say that in the colorectal cancer study, one of the two biomarkers that we finally concluded with, one of them was enriched already from stage 2 of cancer. It was not enriched in stage 1. Among the 4 stages, it started to be enriched in stage 2. You might say, well, stage 2 is already stage 2. You're absolutely right. But when we talked to cancer clinicians, they said stage 2 is already a good place to catch people. A lot of them walk in with much more advanced stages than that. So this is already better than what we have. But of course, the ultimate application of microbiome will be that you catch them already at stage 1. So you can completely turn the cancer around and cure them and get rid of everything. I see that we will get there soon. We have two European consortia working on liver disease, where we study whether microbiome can be predictive of response to therapy, and also whether microbiome can predict progression of late-stage liver diseases. So some of these look very promising right now. Again, it's an ongoing study, but we do see promising results that they can be predictive of disease progression and also efficacy of treatment. And I don't know if you've seen this. There was a paper that was published only a couple of days ago for cancer therapy, for melanoma treatment. Those that were resistant to treatment after giving FMT from healthy individuals, I believe, I haven't read the paper fully, they could actually improve the efficacy of the treatment. So microbiome is definitely doing something. So I believe that prediction using microbiome biomarkers for both disease progression or efficacy of intervention and treatment, I think we are all going to come soon.
Hey,
Thank
Mani.
I think Himanshu has a question. Yeah.
Yeah, hi, Mani. How are
Yes,
you?
Hi, Himanshu. Nice to see you.
Nice to see you after like what, 12 years, right? Hey, nice talk, actually, very nice talk. And like we've been, like as Karthik was mentioning, we've been sort of following, at least I've been on and off following some of your work. So I was sort of wondering, like following up on what one of your last comments, you mentioned that fecal transplant is one of the ways people are treating some of the disease, like Crohn's is a very classic example. But given that nowadays we have access to organoids, and gut organoids are something people are using for a lot of metabolic disorders, especially when you have or even for cancer like colon cancer and all those things. So is it possible to sort of test some of your hypotheses that you have of how microbiome is changing vis-a-vis a normal tissue and a cancer tissue, and is it sort of do to make an organoid and do fecal transplant and see what happens in the host as well? Is it something that is a feasibility at the moment right now, or would it be something that a direction would be taken in that direction, because something that we would like to understand what is happening in host as well, because that's something is probably missing in many of the studies that are being done just with a fecal sample.
It's an excellent question. I believe that fecal transplantation will be and at some point should be replaced by more defined therapy. It can still be complex. It doesn't have to be defined in terms of exactly 100% of the ingredients are defined. It can still be complex, but more controlled. Because in the fecal sample, we have no idea what is there. And we can only measure what is dominant. Like I was saying in an answer to a previous question, we have sequenced a fecal sample with what is that 36 gigabases of shotgun metagenomics, and there were species that were not present or they were not detected with 36 gigabases of sequencing, but there are so many species that actually can be recovered. Outside of if you start culturing them under various conditions. So they're all hidden. And 36 gigabases is a lot. It's bloody expensive. But if you think of ratios of bacteria that goes from 1,000 to 1 or a million to 1, then 36 gigabases is not enough at all. So there are so many hidden microbes that might be doing things that you don't want, and you cannot even detect them because even this sequencing that you cannot detect them. So something more defined, even if it's a microbiome consortium, but a more defined consortium is where I think the future should go to. Fecal transplantation works for now, but it shouldn't be the end. Having said that, for talking about the role of organoids, etc., there is a lot of work going on in organoids and microbiome as well. We have started something where we are trying to, only using gut organoids, trying to figure out how the signal transduction in these layers of gut epithelial cells will be affected if you have different microbes in these organoids. We started working on these. As of now, I haven't seen people using organoids to grow microbial communities and then use them as therapeutic community. I haven't seen that yet, but it's definitely a way forward. If we are thinking of defined communities as a way forward, one way is to say, well, I know exactly what composition it should be. It should be A, B, C, and D at X, Y, Z ratio, mix them together, and then give it to the person. That could work, but I think taking the exact same bacteria in that composition and throwing it onto an organoid where the host cells are already there. It's in vitro, but still they still perform a lot of the work. Letting them interact with them and work on the signal transduction for a while, communicating with the host epithelial cells, and then taking them into a therapeutic product. I can see that as an attractive solution as well. We're not there yet, as in it's not a publicly available solution yet, but there are so many microbiome startups right now, especially in the Cambridge, Massachusetts area. I wouldn't be surprised if somebody
has already done it.
already, and one day they will just come up with a solution. They already have a defined cocktail for Clostridium difficile infection from Serious Therapeutics, and they're working on another cocktail for IBD. So things are happening. It's a very fast-moving field, and we might just be suddenly surprised by an announcement by one of these microbiome startups saying, "Hey, organoid-based microbiome communities
ready
as
to go, right?"
treatment for IBD."
Yeah, just one very quick question. Is there a sense also of trying to understand the kinetics of change of microbiome, like something like Farmington study that has been done where you're like following same cohort of people across ages, across sort of their life? Are there certain studies like that where they're like tracking from infant or maybe even like newborn to like certain age where probably microbiome stabilizes. I don't know if that is anyway a concept that a microbiome stabilizes, but are there studies like that being done as well?
There are not a lot. The initial microbiome studies were all focusing on cross-sectional design. We'll find patients, find healthy controls, just sample them cross-sectionally at a given time point and analyze them. This is how things were done. And this is where we were always plagued by the causality issue, because who knows what came first. The right way to study the role of microbiome will be prospective studies. Start recruiting people and start following them over time in prospective cohorts, and over time, just statistically, some diseases will show up in a fraction of the population in the cohort. Then you can see how the trajectory of the microbiome changed in those individuals compared to those that did not get the disease. The unfortunate truth is that this was realized much later, so prospective cohorts are not more than 6-7 years these days. Prospective cohorts for blood sample and urine sample, and these exist for a long time, but they never collected fecal samples. And these are probably less than 10 years old. And I also know of some studies where kids are followed from birth. One of my postdocs is part of an asthma center where they treat childhood asthma, and they have a toy project which is the Daily Gut Project, and he and his associates they started a study where they sample their own kids every day. Wow. Every single day they collected samples from their kids. I mean, it's difficult to ask somebody to do it, so they wrote an ethical permission for doing it themselves and got it approved, and they are actually doing it. There's 3 or 4 of them. They collected samples every day. This is going to tell us how microbiome is changing in the infants over time. At this resolution, nobody has done it. So more things will come up. And I hope to see more of these infant studies in other places. And obviously, to follow a person's entire life, if microbiome as a field only started 10 years ago, I mean, we need to wait for 80 years to see the first person who was sampled from birth till 80 years, right? So we are way early in that.
Okay, good. Okay. Mani, I'll have to go. I think Karthik has some more questions.
Yeah. No, I think, we are also sort of, I think we've held Mani long enough. So I think we'll probably wind up in 5 quick minutes. So maybe one last question from, I think Bharat and Aarti had a question each, and we should probably wind up with that. So Bharat, quickly, if you can.
Yeah, hi. Karthik and Imanshu, thanks for organizing this, and Dr. Mani, excellent presentation there. Just a brief, Bharat Prithviraj, alumnus of the Rob Knight Lab and also part of the Earth Microbiome Consortia, actually collaborated with Karthik and Imanshu. Excellent talk, really enjoyed that. And I just had one very specific question on your slide with the Parvomonas micra and Fusobacterium nucleatum. And I was curious because has your group had interest in looking at the oral biomarkers for CRC, especially since O'Toole's group had reported Fuso as one of the predictive salivary biomarkers? So just that query. Thanks.
What I forgot to mention, and thanks for reminding me of that, is that every single strong biomarker for colorectal cancer that was validated across the different cohorts in our meta-analysis, they are all traditionally oral microbiome members. They don't have a normal place in the gut microbiome. They can be there because it is still one gastrointestinal tract, so they can get there. But they should be there in very low amount and not even detectable unless you have a very high sequencing depth. But when the patients have cancer, they are all showing up at a reasonably high level. Again, in the healthy individuals, when they were not measured, I would say it was only not detected at the sequencing depth. They were probably also present because most people carry Fusobacterium in their oral microbiome. So our idea, or at least this is how we proposed it in our paper in 2017 in gut, that there's something going on in the oral gut barrier and there is a translocation of the oral microbiome into the gut, which would have been very little given the acidic barrier in the stomach, etc. But apparently during colorectal cancer, that barrier gets weaker, and traditionally oral bacteria end up being seen higher in the gut. This is as far as we can go, and that makes perfect sense that the same bacteria can also be measured earlier in the mouth, because it's they are supposed to be just nicely hanging on to the surfaces in the mouth, but maybe they are going higher in their abundance, and maybe they are also getting detached and free flowing, and maybe the interaction between the immune system in the mouth and these bacteria is also getting weaker, which potentially leads to this oral gut translocation during cancer. This is our gut feeling. We haven't investigated this very thoroughly, especially from the oral microbiome point of view, because we didn't have access to such samples. But of course, Paulo too has been following it for quite some time. So I'm definitely not surprised, and I actually believe what they say is that oral members can also be predictive.
Thank you. Yeah, that's great to hear.
Okay. So the last question maybe, Aarti had a question. Aarti, you there?
Oh, hi. Hi, Dr. Mani. This is Aarti, and thank you for this wonderful talk. Following up on the question which you previously answered on how do you filter organisms or something of that sort. So how
Sorry, how do
exactly
you filter what?
the organisms from the taxonomic profile? There was a question regarding how do you filter out, I mean, you had answered something like E. coli, if it's 90%, how do you remove it, and so on and so forth, right? Yeah. So how exactly do you think you can distinguish rare species from false positives?
Good question. Rare species could be really rare species, or it could be a contamination during your sample preparation, during your DNA extraction, during your sequencing, where there was a bacterium in the air which just got in, or there was DNA in the air which just got in. It's an excellent question. It's very difficult. What will help you is if you saw the same rare bacterium under different experimental conditions, in different sequencing runs, or when experiments were done by different labs. If you see the same rare bacterium multiple times, then it's probably a real rare bacterium rather than an environmental contaminant from this lab versus that. Of course, in the extreme, you can say that, well, all labs in the city are contaminated with this bacterium, then it's difficult to rule that out. Then you will have to see between different cities, between different states. It's a good question. The only way to figure this out is by comparing data generated from independent experiments, independent labs, independent protocols. Because sometimes, there's also contamination, DNA contamination from kits of DNA extraction. So you use the same kit in different places, maybe you will have the same contamination, but then you will have to check with another lab that used a different kit to do the DNA extraction. Or if this is disappearing there, then it probably came from the kit. But if it is still there, then it is independent of the kit. So you will have to rule out these confounding factors one by one for rare bacteria.
Great, Mani. So thanks a lot. I mean,
Thank you.
I think that might be an auto cutoff for the as per the settings. So let me just thank you once again for a wonderful talk. I've been in so many webinars, but I don't think the Q&A went this long in any of them this far. So wonderful talk, really exciting, really engaging. Thanks. We will be in touch. We'll we'll probably meet sometime to discuss if there are possibilities for collaboration and things like that. So wonderful talking to you, yeah, we hope to meet you in person sometime soon.
Thank you. Thank you.
Thank you all. Thank you to all the panelists as well, and the participants. Thanks, Mani. Have a great evening. Yeah.
Thanks. Bye.
Bye-bye. Thank you.